The process of aerobic cell respiration is a very complicated and a very intricate process because it actually consists of many individual processes and many individual steps. So far we discussed glycolysis, pyruvate decarboxylation, and the citric acid cycle. Now, the final process of aerobic cell respiration, the culmination of aerobic cell respiration, is a process we call oxidative phosphorylation that takes place on the electron transport chain of the inner membrane of the mitochondria. And this is what we're going to introduce in this lecture. But before we focus on the electron transport chain and oxidative phosphorylation, let's remember what happened in glycolysis, pyruvate decarboxylation, and the citric acid cycle. So in glycolysis, we broke down glucose into pyruvate molecules, and we also formed NADH molecules and a few ATP molecules, and this took place in the cytoplasm of the cell. Now, in the presence of oxygen, the pyruvate molecules will be moved into the matrix of the mitochondria. And in the matrix, we're going to prepare the pyruvate molecules for the citric acid cycle by a process called pyruvate decarboxylation. So we form acetyl coenzyme A molecules. And those acetyl coenzyme A molecules are then moved into the citric acid cycle, which also takes place in the matrix of the mitochondria. So in the cytoplasm, we have glycolysis, but inside the matrix, we have the citric acid cycle and pyruvate decarboxylation. Now, the entire point of the citric acid cycle is to actually further oxidize the glucose derivative molecules so that we can extract those high energy electrons and place those high energy electrons onto special carrier molecules we call NAD+ and FAD. Now, what's the point of extracting these high energy electrons? Well, the point of extracting these high energy electrons is to use the high energy electrons to basically oxidatively phosphorylate ADP molecules into ATP molecules along the electron transport chain. So we ultimately use the NADH molecules and the FADH2 molecules produced in glycolysis and the citric acid cycle to actually generate those high energy ATP molecules, and this takes place in the electron transport chain. So the culmination of aerobic cell respiration is oxidative phosphorylation along the proteins of the electron transport chain. So the NADH molecules formed in glycolysis, which takes place in the cytoplasm, and the NADH and the FADH2 molecules formed in the citric acid cycle, which takes place in the matrix of the mitochondria, are basically used to ultimately reduce diatomic oxygen molecules to form water, in the process also generating high energy ATP molecules. And this is shown summarized in the following diagram. So this is a single mitochondrion. In the mitochondrion, we have several compartments. We actually have two different membranes. We have an outer mitochondrial membrane and the inner mitochondrial membrane. And this electron transport chain is found on the inner mitochondrial membrane that consists this folding pattern known as cristae that we're going to focus on in just a moment. So let's zoom in on a small section of the inner mitochondrial membrane. This is what we're going to find. So this is the matrix, this portion, and this is the intermembrane space, this space between the inner membrane and the outer membrane. So the outer membrane is somewhere right here. So as we can see from the diagram, our electron transport chain is located on the inner mitochondrial membrane, and it consists of these special types of proteins, as we'll discuss in a future lecture. So we have protein complex 1, protein complex 2, protein complex 3 and 4, as well as other proteins, as we'll see in a future lecture. And we have this final protein molecule known as ATP synthase. So ultimately what happens is protein complex 1 is responsible for accepting those high energy electrons from NADH molecules produced in glycolysis and the citric acid cycle. And we extract these high energy electrons into this area here. At the same time, we also regenerate those NAD plus molecules needed for glycolysis and the citric acid cycle to actually continue taking place. Now, the FADH2 molecules produced in the citric acid cycle are actually taken by protein complex 2. Now, once these high energy electrons are extracted, they begin to move along the proteins of the inner membrane of the mitochondria. And from basic physics, we know that whenever electrons flow from point 1 to point 2, the flow of that electrons, or the flow of the electrons, creates an electrical current, and that electrical current can be used to carry out some process. So what process are we ultimately carrying out? Well, as these high energy electrons are moving along the proteins of the electron transport chain, that allows some of these proteins to actually act as proton pumps and pump H+ ions from the matrix of the mitochondria to the intermembrane space. So protein complex 1, 3, and 4 actually allow the movement of these H+ ions. So the movement of this electric current allows us to actually pump these H+ ions to the intermembrane space of the mitochondria. And once these electrons end up on protein complex 4, this is where we actually use those electrons to actually reduce the diatomic oxygen, the same oxygen that we breathe in from the environment, to form the water molecules. In the process, once we generate this proton gradient across the two sides of the membrane, we use another protein complex known as ATP synthase to actually generate those ATP molecules. So ultimately, we establish a proton gradient in which we have a high proton concentration here and a low proton concentration here. And this ATP synthase basically uses that established electrochemical gradient of H+ ions, moves these H+ ions down that established electrochemical gradient from a high to a low electrochemical potential, and that allows the ATP synthase to actually phosphorylate ADP to ATP. And that's why we call it oxidative phosphorylation, because we actually use an oxygen on the electron transport chain. So remember, the citric acid cycle doesn't actually require oxygen itself, but the electron transport chain does. And we need the citric acid cycle to generate the majority of the NADH molecules and these FADH2 molecules. So the electron transport chain is a series of proteins that receive these high energy electrons from NADH molecules and FADH2 molecules, and they move these electrons along the inner membrane of the mitochondria. And the final acceptor of these electrons is oxygen. So we reduce oxygen to form the H2O molecules. Now, we know from physics that as these electrons flow, they create an electrical current, and that basically allows us to carry out some type of process to do work. And so we are able to actually pump these protons to establish a proton gradient. And that proton gradient is then used by ATP synthase to actually generate those ATP molecules. So we can summarize these 4 steps in the following 4. So 1, 2, 3, and 4. In 1, those high energy electrons are extracted from these high energy electron carrier molecules synthesized in the citric acid cycle and glycolysis. And once these electrons are extracted, they move along the proteins, the enzymes of the electron transport chain. And that ultimately allows some of these proteins to actually pump the H+ ions to the intermembrane space, the space between the inner and the outer membrane of the mitochondria. And this establishes an electrochemical gradient for the hydrogen ions. So this takes place here, here, and here. Then the electrons are ultimately accepted by that diatomic oxygen to form water molecules. And in the final step of the electron transport chain, the fact that we have An unequal distribution of H plus ions basically means we have an electrochemical potential difference between the two sides of our membrane, and that allows the ATP synthase to actually use that energy that is stored in that proton gradient to generate these ATP molecules. So we conclude the following. The oxidation of the fuel molecules in glycolysis and the citric acid cycle is actually coupled to the oxidative phosphorylation of the ADP to ATP by using this oxygen in protein complex 4 by the establishment of this hydrogen ion gradient on the electron transport chain. So what we're ultimately doing in aerobic cell respiration is we're transforming one form of energy into a different form of energy, into a form that we can actually use by the cells of our body. So we basically transform the energy that is stored in the movement of electrons into the energy that is stored in the gradient that exists between the two sides as a result of the unequal distribution of H plus ions. And then we ultimately transform that energy into the energy that is stored in the chemical bonds of the ATP molecules, because it's the ATP molecules which can be used by our cells to carry out a variety of different types of processes. So the final thing that I'd like to focus on in this lecture is some facts that you should know about the mitochondrion found inside our cells. So as I mentioned in the beginning, the mitochondrion actually consists of two different types of membranes. We have the outer membrane and the inner membrane. So the innermost compartment of the mitochondrion is known as the mitochondrial matrix, and the space between the two membranes is called the intermembrane space, and this is found entirely in the inner mitochondrial membrane. Now, there are several important differences between these two membranes. First of all, notice that the inner membrane actually consists of these folding patterns, and the folding patterns create something called the cristae. Now, cristae are these inner foldings that greatly increases the surface area on which the electron transport chain can actually lie. And so we have a greater surface area as a result of these inner foldings, and so that means we can have many more of these electron transport chains, and that greatly increases our cell's ability to actually undergo oxidative phosphorylation. Now, the second important difference between the two membranes is the fact that the outer mitochondrial membrane contains these special mitochondrial pores, these special mitochondrial proteins called porins. Mitochondrial porins, also known as VDAC, where VDAC basically stands for voltage-dependent anionic complexes. And so what happens is these mitochondrial porins found on the outer membrane basically allow the movement of anion molecules, so small molecules which are anions. So things like chloride ions, things like ATP molecules can actually move across the outer mitochondrial membrane from the cytoplasm into the intramembrane space without much difficulty. But the inner membrane doesn't actually contain these mitochondrial porins. So that means it is impermeable to charged molecules, and it's impermeable to polar molecules. So molecules such as citrate molecules or pyruvate molecules or ATP molecules actually depend on special protein membranes or membrane proteins to actually shuttle those molecules across the matrix and into the intramembrane space. So the outer membrane is permeable to the majority of small molecules and anions due to the presence of voltage-gated channels we call mitochondrial porins, or VDAC. It allows the movement of anionic metabolites such as chloride molecules and ATP molecules and so forth. But the inner membrane lacks these mitochondrial porins and so is impermeable to ions and polar molecules. So metabolites such as pyruvate molecules or ATP molecules or citrate molecules actually depend on special protein transporters to move across the two sides of the membrane. Now, if we take a small section of this region here, this is what we're going to get. So this is the outer and the inner membrane. Now the inner membrane, on the matrix side, we have a negative charge. On the intermembrane space side, we have a positive charge. And that's why this side of the membrane is called the N side, N for negative, and this side is called the P side, P for positive. Likewise, this N side is also sometimes called the matrix side, while this side is sometimes called the cytoplasmic side, because the intramembrane space is pretty much connected to the cytoplasm via these mitochondrial porins found in the outer membrane of the mitochondria. And as I mentioned just a moment ago, the folding pattern of the inner membrane, this structure here, consists of these cristae, which greatly increase the surface area on which oxidative phosphorylation can actually take place.